Welcome to our presentation on interpreting biomedical data and turning complex biological information into actionable medical knowledge. Over the next slides, we will explore the challenges, tools, and future directions that make precision healthcare possible. This opening sets the stage for why converting raw datasets into clinical insights is critical for better patient outcomes and scientific discovery. Biomedical research in healthcare are producing data at an unprecedented rate, from sequencing reads to high-resolution medical images, creating both opportunity and overwhelm. In this section, we introduce the idea of a data explosion and why managing volume, variety, and speed is now a core challenge for clinicians and researchers seeking meaningful signals in noisy datasets. Modern biomedicine generates massive datasets that span genomics, proteomics, imaging, and clinical records, and these layers create both rich context and analytical headaches. Managing volume, variety, velocity, and veracity requires new frameworks, while breakthroughs like whole genome sequencing show how terabytes per person can revolutionize personalized medicine and our understanding of disease mechanisms. Moving from molecules to medicine means integrating multi-omics data to reveal a fuller biological picture, connecting molecular mechanisms directly to clinical outcomes. This slide highlights how combining genomic, proteomic, and metabolic profiles can help trace pathways from cellular events to patient symptoms, enabling more targeted hypotheses and ultimately informing therapeutic decisions in the clinic. Interpretation matters because raw numbers are meaningless without context and expert analysis, and accurate interpretation is the engine of precision medicine. When we put data into rigorous frameworks, we turn measurements into tailored diagnoses and treatment plans, but misinterpretation risks biased results, invalid conclusions, and, importantly, harm to patients if decisions rest on faulty inferences. This section outlines the foundations of biomedical data interpretation and the core principles that underpin reliable analysis. We will cover the basic building blocks, data types, pre-processing standards, statistical thinking, and computational approaches that together create a trustworthy pipeline from raw signals to clinically relevant insights. Biomedical data appears in many forms: genomic sequences and proteomic profiles, clinical time series, and advanced imaging scans, each bringing unique biological information. Equally important is format variety, from spreadsheets to multidimensional arrays, and the need for careful pre-processing such as cleaning, imputation, and normalization to ensure analyses start from a sound, comparable baseline. A robust toolbox combines visualization, machine learning, and statistical methods to extract meaningful patterns from complex datasets. Visual techniques like scatter plots, heat maps, and PCA make structure apparent, while classification algorithms such as k-nearest neighbors, decision trees, and support vector machines identify predictive patterns, and practical examples show how these methods reveal biomarkers hidden in high-dimensional data. Principal component analysis helps by transforming high-dimensional biomedical datasets into lower-dimensional representations that highlight the most significant sources of variation. By reducing noise and complexity, PCA can expose biological patterns that were previously obscured, for instance revealing critical cardiac biomarkers in myocardial infarction datasets that inform prognosis or therapy selection. Artificial intelligence and machine learning are transforming how we interpret biomedical data by automating pattern discovery and enhancing predictive power. In this section, we look at the ways advanced models augment human expertise, from uncovering subtle correlations in large cohorts to accelerating image analysis, ultimately enabling faster, more accurate insights that support clinical decision-making. AI plays three complementary roles in biomedical analytics: discovering hidden patterns across vast datasets, powering deep learning models for complex tasks like image interpretation, and enabling clinical applications that predict outcomes or identify tissue abnormalities. When trained and evaluated carefully, these tools can generate hypotheses, assist diagnosis, and surface interactions at scales beyond traditional methods. This case study shows how a specialized convolutional neural network can classify structural MRI scans to assist autism diagnosis, demonstrating technical innovation and clinical promise. Post-hoc visualization techniques then highlight abnormal brain regions for clinicians, and the broader implications suggest similar approaches could extend to Alzheimer's, Parkinson's, and other neurological disorders for earlier detection. Interpreting biomedical data comes with significant challenges that span technical, ethical, and organizational domains, and addressing these barriers is essential for reliable outcomes. In the following slides, we'll explore data quality and bias, interoperability issues that block collaboration, and practical steps researchers and clinicians can take to improve reproducibility and trust. Data quality problems like missing values, measurement noise, and patient heterogeneity hinder accurate analysis, while ethical challenges around privacy, algorithmic bias, and transparency demand careful governance. Additionally, gaps in standardization slow progress towards truly effective precision medicine, making it essential to prioritize robust datasets, fair models, and clear policies to protect patients and ensure reproducible science. Collaboration is often stymied by data silos where clinical and research datasets remain isolated across institutions and disciplines, preventing comprehensive analysis. To overcome this, we need shared integration frameworks that combine genomics, imaging, and clinical sources, and collaborative solutions, such as interoperable digital twins, can enable holistic patient modeling and accelerate collective discovery. This practical tools section introduces approaches that make analysis reproducible, scalable, and collaborative, focusing on software stacks, cloud-based notebooks, and evaluation practices. Emphasizing hands-on platforms and workflows helps teams move from exploratory analyses to validated findings, ensuring that methods are transparent, repeatable, and ready for translation into clinical research or operational settings. Open-source Python libraries have democratized biomedical analytics by offering accessible, powerful tools for data processing and model development. Collaborative environments like Google Colab and Cloud Notebooks enable teams to reproduce and share work seamlessly, while advanced visualization and model evaluation tools empower researchers to interrogate results critically and identify biases before clinical application. Emerging trends point to large language models for biomedical text mining, AI-driven signal analysis for continuous monitoring, and tighter clinical integration where models support point-of-care decisions. These directions promise faster extraction of insights from literature, improved detection of arrhythmias or psychiatric signals, and real-time decision support embedded in clinical workflows to assist physicians when it matters most. Looking ahead, the future of biomedical data interpretation will be defined by close collaboration between human expertise and artificial intelligence to transform healthcare delivery and accelerate discovery. By combining domain knowledge, robust analytics, and ethical practice, we can build systems that augment clinicians, support earlier diagnosis, and translate complex data into tangible improvements in patient care. To conclude, interpreting biomedical data is the key to unlocking precision medicine's promise, but it requires statistical rigor, AI innovation, and ethical responsibility working together. Cross-disciplinary collaboration will accelerate breakthroughs and improve outcomes worldwide. So let's embrace these tools to convert complex data into life-saving knowledge and better health for all.